Hello, everybody, and welcome back to Creation Myths. Today, we are doing another round of responding to critics, and the critic to whom we are responding is once again Dr. Casey Luskin from Discovery Institute. This is going to be another round in our ongoing conversation on junk DNA and related topics. This came about because Dr. Casey Luskin from Discovery Institute Center for Science and Culture appeared on the Christ Jesus Ministries Current Topics in Science podcast, specifically the episode from July 13th, 2024. In this interview, Dr. Luskin talked about junk DNA and specifically, at the time of this recording, our recent debate on junk DNA from May of 2024. So in this video, I'm going to go through a few segments from this interview and point out a couple of errors in Dr. Luskin's arguments and statements, as well as a pretty serious misunderstanding of my position regarding junk DNA and how we can characterize genomic regions as junk DNA, like how we actually know that a region is junk, that it doesn't have a specific function. Now, I'm going to say this again at the end, but I want to make sure it's crystal clear before I go into any of this. I don't think Dr. Luskin is trying to deliberately strawman my arguments. I think this is a good faith misunderstanding of the case that I am making that the human genome is largely junk and how we know that. Okay, so I want to make sure I say that up front. So the first clip here is pretty short and it's a small point, but it is worth addressing. So check this out.
The quote unquote junk DNA, the DNA in our genomes that does not code for proteins, is actually very important for performing various biological functions. And just sort of to frame the issue here, for many decades, it was thought that the vast majority of our genome, say 98% plus, was what we call junk DNA. Basically, it was DNA that did not encode for proteins. And the assumption from an evolutionary point of view was that DNA was basically useless.
Okay, so this claim, the idea that we, biologists, for decades thought that 98% plus, I think that was the phrase, 98% plus of the genome was junk DNA. This is the idea that all non-protein coding DNA was thought to be junk. And that is not true, and that has never been true. The reality is that the term junk DNA was first defined in the literature in 1972. And by then, we had documented a whole lot of functional non-protein coding DNA. Okay, we knew about tRNAs, ribosomal RNA, promoters, centromeres, telomeres. So it has never been the case that the term referred to all non-protein coding DNA. It's just historical fact, okay? Now, Dr. Luskin isn't a biologist. He's a geologist and a lawyer by trade, so he may not be familiar with the history here. However, other people affiliated with Discovery Institute, such as Drs. Jonathan Wells and Douglas Axe, are biologists and ought to know better. They should know the history, but they make this claim anyway, as I recently documented in a short video on this from December of 2023. For them, if I'm being honest, I think they're lying. I think they're being dishonest about it. It's their field, they're biologists, they're talking about this specific thing. Either they ought to know the history behind it so they don't make erroneous claims, or they do know the history and they're purposefully making the inaccurate claims. Now, I don't think that's the case for Dr. Luskin. I think he probably heard or was told this claim and basically said, okay, and repeated it, right? Because again, he's not a biologist. So Dr. Luskin, I don't mean to denigrate your expertise. It's just you're not in the field. You're not immersed in it like working biologists would be. So that's what I think is going on here. This next clip I'm going to show you is also relatively short. So we'll address that. Then we'll get into kind of the meatier part of this video.
So what I would say with pseudogenes is, yes, it's very well established that they can have functions. In fact, it's well established why pseudogenes even often have to resemble protein coding genes, even though they don't produce a protein. Many pseudogenes will produce an RNA transcript. That transcript is not capable of being translated into protein, but that transcript is then capable of going out into the cell and regulating the protein coding version of that gene. So you basically have these RNA transcripts from the pseudogenes and RNA transcript from the protein coding version, they can interact in the cell, and that can be a way that they can regulate the actual usage of the protein coding version of the transcript. And you need those similarities between the pseudogene and the protein coding version to be there, because those similarities allow those transcripts to then interact properly and allow them to actually regulate the expression of the gene.
I find this particular section of that interview really interesting. This discussion of pseudogenes and how they would be functional. Basically, what Dr. Luskin is saying is that the pseudogene sequences require sequence specificity. So if the pseudogene doesn't match the gene it's regulating, the target RNA, then it's not going to be able to do that function. That is true, right? This means that this function requires sequence specificity. And it is also true that the vast majority of pseudogenes don't experience purifying selection to match the target protein coding sequence. Right? That is just a fact. Most pseudogenes are just accumulating mutations without selection operating. They're accumulating mutations without a fitness cost. That means that there's no selection maintaining that match to the would-be target sequence, which means most pseudogenes aren't doing this function that Dr. Luskin is describing. So while I think largely factually accurate, this little clip of the interview right here, this actually undermines Dr. Luskin's position on the functionality of pseudogenes and junk DNA more broadly. He's correctly identifying potential functions that require sequence specificity and describing why they require sequence specificity. The problem is that these sequences do not experience purifying selection, so they aren't maintaining sequence specificity, so they aren't doing these functions. And that's true not just for pseudogenes, but for the vast majority of the human genome, whether it's pseudogenes, endogenous retroviruses, retrotransposons, whatever the case is. So now I want to jump ahead a bit to the part where the host specifically asks a question regarding my debate with Dr. Luskin on junk DNA. We're going to watch the question that the host asks, and then we're going to go through Dr. Luskin's answer. So here's the question, then we'll talk about stuff.
For this next question, we're going to take a look at your recent debate on junk DNA with Dr. Dan on the Non Sequitur show. Dr. Dan argued against the idea that the endogenous retroviruses, or the ERVs, can be explained within the ID scientific model. He gave 3 reasons for his argument, which goes as follows. Will this piece of RNA from the virus hybridize, form a double helix, with this piece of RNA from your genome? And we can show, based on the biophysics of it, that the answer to that question for most endogenous retroviral sequences is no. Because, 1, the sequence is not transcribed because the LTRs are broken down. 2, the transcribed sequence is too short to matter for the length of that complete virus. And 3, the sequence is diverged from the virus by too much, and it would no longer effectively hybridize with it within the cell. So Dr. Luskin, is Dr. Dan correct, or do ERVs fit within the design model?
I want everyone to keep that question in mind as we go through Dr. Luskin's answer, because we're going to point out a number of things about that answer. But one thing I want you to ask yourself as we listen to Dr. Luskin respond is this question. Does he answer that specific question at any point? Does he answer the question that was asked? So now let's watch his answer.
During the debate, Dr. Dan really, I think, exemplified the kind of exact reasoning that we're talking about here. That what evolutionary biologists tend to do is they tend to assume that if we don't understand what something is doing, that therefore it is functionless junk and is doing nothing for you and is evidence for sort of some, random garbage accumulating in the evolutionary past, all right? And Dr. Dan exemplified this kind of reasoning over and over and over again in our debate. I even predicted, if you want to talk about predictions, I predicted at the beginning of the debate that we would see that Dr. Dan was going to show us how evolutionary reasoning basically hinders and discourages people from studying whether or not the junk is functional, because they just assume that it can't do anything, because they can't envision what it might do, so they assume it's junk.
All right, let me stop here. That's the opposite of what I did. I even said this during the debate. I explained up front that we're characterizing things as junk based on what we know, based on what their sequences are, and what those sequences can or cannot do. And I'm going to, again, be very clear here. What I'm saying about these various proposed functions is this. Most members of most classes of DNA, right, ERVs, retrotransposons, pseudogenes, are incapable of doing the functions attributed to some members of each of those classes. You cannot extrapolate or assume that they're all doing this or that function just because a minuscule number have been documented to do a specific function. And the reason for this is that specific functions require specific sequences or specific activities. For example, going back to the endogenous retroviruses, if the purported function is to match the sequence of a real virus and interfere with it, then the ERV RNA has to actually match the sequence of the viral RNA, or it has to be complementary to the viral RNA. If it doesn't match enough for those two pieces of RNA to hybridize, right, to form a double helix, which is something we can calculate based on those sequences, then it's not doing that particular function. We can directly answer the question, can this piece of RNA bind to that piece of RNA? If the answer is no, then it's not playing an antiviral role. Does an ERV sequence bind to transcription factors? Yes or no? If no, it's not an enhancer. These are questions we can, and in a great number of cases, have answered. And those answers are what enable us to say a specific region of the genome is not doing these specific proposed functions. Might it be doing something else? Sure, provide evidence for that. It might be doing something else is not evidence, it's conjecture. And what's happening here is the anti-junk advocates are getting out over their skis and jumping to conclusions where the data do not warrant it. Back to Dr. Luskin.
And the argument that you just described here, that basically we have certain repetitive DNA, he's talking about ERVs, which are so short that they can't be transcribed, or they can't actually perform the function that they would normally perform because they're broken down. He said that they are quote unquote degraded. All right. And so the idea is, well, what does it, in sort of, if it's actually a virus, what does an endogenous retrovirus do? Well, it will produce certain proteins, certain enzymes. Those enzymes then allow it to form this retrovirus, which can then basically go out into your genome and retrotranspose and reinsert the viral DNA back into another part in your genome. Okay. In Dr. Dan's mind, if an ERV isn't doing that function, then there's no other function it can do. All right. That was the way he argued, that basically if it can't produce the enzymes required for retrotransposition, that's all it does. There's nothing else it can do.
Okay, that is completely 100% wrong, and I genuinely do not understand how Dr. Luskin could think that's my argument here. Not based on what I've said, and not based on the question he's, in theory, answering. The argument is not that if an ERV can't do retrovirus things, if it can't retrotranspose, it can't be functional. That's not my argument. That's not the argument of mine he just heard from the interviewer. My argument is that we have specific functions proposed for ERVs. Antiviral activity, for example, as the interviewer explained, and for very specific reasons, most ERVs cannot do those functions. That's what he's supposed to be addressing. Instead, he just presented, purposefully or not, knowingly or not, and I think not on purpose and not knowingly, an egregious straw man of my position. And again, I don't think it was on purpose. But at no point before, during, or since our debate have I said or conveyed the idea that if an ERV is not doing its ancestral functions, it can't be functional. I mean, obviously that's not true. We've co-opted viral promoters. We've co-opted viral genes. Neither of those functions require retrotransposition. My argument is that for the vast majority of that 8% of our genome that's endogenous retrovirus, the specific sequences are such that they can't do any of the observed or proposed functions. And I've provided quite detailed technical reasons why that is the case. If they don't match the would-be target virus closely enough, they're not doing antiviral activity. If they can't bind transcription factors, they aren't acting as enhancers. If they aren't transcription start sites because the LTRs have experienced deletions, they can't be promoters. The interviewer accurately conveyed some of those regions, specifically with regard to the antiviral activity. And Dr. Luskin is responding to a completely different question. I continue to believe this is a good faith misunderstanding, so I hope I've done a better job of explaining my position here than I've done in the past. I also think there could be some confusion here because of stuff we talked about in our debate. In our debate, we talked a lot about specific elements called lines, which are a type of retrotransposon, and in some ways they're pretty similar to endogenous retroviruses. Specifically for lines, at least some of the proposed functions, and I think some of the functions that have been directly observed, are based on their ability to carry out at least part of the process of retrotransposition, where they cut themselves out and then paste themselves in somewhere else in the genome. And specifically, this would be their participation in double-stranded DNA break repair. In that specific case, that very specific case, the inability to retrotranspose does preclude that specific function, right? If they don't have the integrase enzyme, they can't do that specific thing. But I want to be crystal clear. The absence of that activity only precludes that specific function. Inactive lines that do not retrotranspose, which is almost all of them, could still be, for example, promoters. And to evaluate that activity, we need to use a different set of criteria. I think it's possible that's where some of the wires are getting crossed here. So if that's the case, Dr. Luskin, I hope that clarified things. So now I'm going to skip over a short bit, about a minute in Dr. Luskin's ongoing answer. He talks a bit about the origin of endogenous retroviruses, and it's not relevant, this part. So I'm just going to skip ahead a little bit to the last section I want to talk about. Here you are.
But let's talk about Dr. Dan's specific challenge. Do we know, he actually talks about very short fragments of ERV, of what we think are ERVs, that are too short and that don't actually produce any RNA transcript and are too short to do anything else. The question is, do we know of functions for ERVs that fall into that category? And the answer is yes, okay? We do know of pretty much any time Dr. Dan would claim that we don't know of any function for this or that, he was actually wrong. And we do know a function for many, many types of junk DNA. We actually put up an article on our website documenting over 50 papers that describe function for repetitive DNA that Dr. Dan claimed was too degraded to have a function, okay? But in this particular case, let's just give one example of one function, all right? Enhancers. Enhancers are segments of the human genome that are involved in regulating the protein coding parts of the genome, okay? They're involved in regulating genes. And what's interesting about enhancers is you can have an enhancer that regulates a gene that could be very, very far away in the genome. It might even be on a different chromosome. So you could have this repetitive ERV DNA that is on one chromosome that is in fact regulating the expression of a gene totally far away in a different chromosome in the genome, all right? And enhancers can in fact be very short. I looked this up before our interview. You can have enhancers that are as short as 50 base pairs. And I'm sure that that's, probably some of the examples of ERVs that Dr. Dan thinks are too short or even longer than that, much longer than that. So you can have ERVs functioning as enhancers that are very short, and they don't need to provide a transcript, by the way, to do this enhancer function. You can have other regulatory proteins latching onto an enhancer, which then are involved in a complex regulatory system to regulate that protein coding gene somewhere else in the genome. So enhancers can be very short. They can be part of repetitive DNA, like ERVs, and they don't have to produce a transcript, and they can have functions involved in regulating gene expression. So, exactly what he said cannot exist, does in fact exist. And I don't know what else to say. I mean, he would make this kind of argument over and over and over again. And he would say that, this type of DNA can't be functional. This type of DNA can't be functional. And we would then come up with examples of no, actually it can be functional. So let's just not keep making this evolutionary argument that assumes that if you don't know what it's doing, that therefore it must be junk.
Okay, so two things are going on here. There is the specific argument related to ERVs that Dr. Luskin gets wrong, and there's the big picture argument that he seems to completely misunderstand what my argument actually is, and as a result of that, repeatedly mischaracterizes me. And again, I want to emphasize, I think this is a good faith misunderstanding here. So first, the small thing. Related to ERVs, he's proposing antiviral functions and gene regulatory functions. These functions all require specific sequences in the form of either being compatible with the target virus or binding specific transcription factors or initiating transcription, right? And we can look at each individual ERV sequence and say, can this sequence do this job? Can this sequence do that job? And the answer for virtually all endogenous retroviruses in the human genome is no. With vanishingly few exceptions, ERVs do not experience purifying selection, which means there's nothing keeping those sequences the same. They accumulate mutations at whatever the background mutation accumulation rate is. So over time, they diverge from their ancestor viral sequences. They lose the ability to bind to them. Their promoters can no longer initiate transcription. And in the vast majority of cases, they've experienced large deletions, so most of the ERV sequence isn't even there to do a job the intact sequence could potentially do. Dr. Luskin was asked to respond to that argument. Go back and listen to the last few minutes of his answer here. Did he at any point answer the specific question that was asked? No. He responded to a different argument, one which I have not made at any point and is in several important ways the opposite of the argument I did make. Now, bigger picture, there are two lines I want to point out for you. First, in that clip I just showed you, he said,
pretty much anytime Dr. Dan would claim that we don't have any function for this or that, he was actually wrong. And we do know function for many, many types of junk DNA.
And then later, towards the end of the clip I just showed you, he said this.
He would make this kind of argument over and over and over again. And he would say that, this type of DNA can't be functional. This type of DNA can't be functional.
This is the exact opposite of what I've said repeatedly, including in our debate. This, I genuinely don't understand how he's misunderstanding me. Watch this clip from my opening of our debate. I'm also not arguing that we don't find function across all different types of DNA, all different classes of DNA, right? So I'm not saying that just because something is an endogenous retrovirus, for example, that means it's non-functional. You often hear something like, we've documented function in every class of DNA in the genome. And that's true. But if we're talking about, is the genome largely junk DNA, the amount of that function matters. And for most members of most classes of DNA, we actually have direct data that show that they're non-functional. They're actually junk. And I'll give some examples of that as we go. To be perfectly clear, my position is that for basically every class of DNA, we have examples of function. But most members, the vast majority of members of each class of DNA, again, things like ERVs, pseudogenes, retrotransposons, those are classes of DNA. The vast majority of members for each of those classes are unable to do the documented functions that are present in a minuscule number of the members of each class. And the reason they can't do that is because of their specific sequences. So if you're going to claim that they're functional, you need to propose a function and provide evidence for that function. Since the vast majority of these regions are not sequence constrained, there's no purifying selection operating and they accumulate mutations at the neutral rate, those functions must be sequence independent. Why? Because if they had critical functions that were sequence specific, purifying selection would be operating to maintain those specific sequences. Since it isn't, we can eliminate from consideration any functions that require sequence specificity. Dr. Luskin and anti-junk advocates more broadly have not specifically addressed this point. So that's where I'm going to leave this response. Like I said, I think this is a good faith misunderstanding of my argument by Dr. Luskin, not a deliberate misrepresentation, and I have to take some responsibility for that. Obviously, I could be doing a better job conveying my specific argument. I will also note that I've conveyed this to him via email, and it's possible we're going to have another conversation on this in the future, but everyone's busy and it's tricky lining up schedules, so we'll see. But I have made these concerns known to Dr. Luskin independent of this video and before this video. And also I am hopeful that we are going to have another opportunity to talk about it. So that's where we're going to leave it for today. Thank you, everybody. Please, before you leave, hit like on this video, subscribe to the channel if you're not already subscribed, and I want to thank all of my channel members, especially my second-tier members. If you join the channel as a member at any level, you get immediate access to all of my pre-recorded videos when I upload them. You don't have to wait for the public release, which is usually going to be on Wednesday nights in the evening, Eastern time for the United States. So thank you all for watching. Hope you enjoyed this. As always, don't get fooled.